So the key thing is, how do you combine all this data? And that's been a huge question for as long as people have been doing science, right? I have different groups doing different research. Maybe they're doing it on the same project. How do you combine all that data? So I'm going to give you there's many different ways to do it, but I'm going to kind of tell you some of the main ways and then how we did it. So typically, say in an array experiment, you're measuring different, hybridization of different mRNAs to say a matrix. You'll have fluorescent units based on, the more fluorescent, the higher that gene. Maybe it goes from 300 to 1250. But on another one, you might be doing RNA-seq, different scales. So what you can do is you can basically take all that data on the different experiments and you basically scale it so that the highest value is about 100 or 100, and then the lowest value, say, is 0. And you would do that with all your experiments so that, say, in another experiment, we had, our units were, maybe highest one was 20, that would then become 100. So everything, you're able to basically compare everything together. What is also a very popular way to normalize is to normalize to relative to an internal metric. A lot of these studies, and most studies actually, have a control. This would be, say, healthy individuals or healthy mice compared to, say, a particular disease. So what you can do is you can take your disease samples, divide it by your control values, and then what you do is you come up with a fold change. So in this case, you could say, hey, we did this experiment. This gene was 28 times greater than this gene, right? Or less than. So again, this is a way to compare different studies. You have your internal control. This is normal animals. What does the gene do relative to that? Well, what we did in our study was kind of a combination of both. So as I'm going to talk to you about these biosets, they're basic controls or comparisons. So in this instance, we have viral infection versus mock. So these are mice. We find the fold change, right, for each one of those. And every bioset can have more than one. Actually, most of them have more than one comparison, right? You can have control versus 1 hour or 1 day post-infection, 3 days post-infection. So you've got a lot of comparisons into one study. So what we'll do is we'll take the fold change of that, and then what we do is scale the fold change. So all that fold change information is in the same range. So the highest value we can possibly have in our scaled values is 100, and the lowest value is negative 100. And again, this is relative to a mock infected animal or healthy individual or healthy animal. So in our final data set, this gives you kind of example. This is just one time point, but you can see each one of these bars would be a different experiment or would be a study comparison within a different study. The height of that bar would indicate the magnitude of change compared to controls. Anything green is going down compared to control, and anything red is going up. And again, these are the scores that we get. What I want to point out, though, is for a gene to be considered differentially expressed, we use an absolute fold change of greater than 1.2 and a p-value of less than 0.05. So in our final data set, we ended up with 15 studies, basically containing 74 biosets within these studies or comparisons to normal. Within these studies, there was about 300 infected mice and 234 control mice. And again, I didn't touch these mice. I didn't do anything with these mice. I didn't have to house them. I didn't have to do any of that stuff. Somebody else did that, all the experimental information, and I get to use that, for free, basically. So we took all this information, about 10,000 genes changed at least one time point, and about half of them went up and about half of them went down. So when so what kind of variables are in these studies that we got? Well, first off, we got to figure out, what are the factors that actually affect the response to this virus in humans? Definitely exposure and time. The longer or the more virus you get at the beginning, or the longer or shorter or longer after infection. Age plays a huge role in COVID-19. We also have gender depends on gender will affect how you respond to the virus. Certain lung problems, heart diseases, lots of comorbidities will make you more susceptible to the virus or have a worse response. And again, weakened immune system, chronic kidney or liver disease. So in our study, these are the biosets that we have. Again, remember there's 74 biosets. The numbers are given here. In our study, we have time. So days after post-infection, we have 4 time points or 5 time points, half a day, 1 day, 2 days, 4 days, and 7 days after infection. We also have age. You can see the age groups up here. We have young, which is less than 10 weeks old, an adult group, 20 to 23 weeks old, and then we have an age group, which is 52 weeks old. You will notice, and what's nice is just like the human condition, we don't have any samples in the age groups from 4 to 7 days post-infection. These mice die. They don't live that long because they're more susceptible to the virus, just like in humans. So there's no samples here, although there are some in the age group or the adult and the young. And then we also have different viral strains. And given here, these are the different viral strains used in these experiments. They go from less severe to more severe. This bat SRBD is basically a coronavirus. They engineer not to be able to infect cells. So this is kind of our positive negative control in a way. And then everything else would be more severe viral strains as we go up here. And we also have, a lot of these experiments have different initial viral doses. So these are plaque forming units here on the right. The more plaque forming units, the more severe the virus. So you would see, as you can see here, most of our samples use the most or the highest concentration of initial viral dose, and I would say most of our samples are using the most severe form of the disease.